In this video, we're going to look at protein evolution. We're going to see how you can compare sequences of different proteins to look at the evolutionary relationships. We're going to see how protein families have evolved and diverged, and we're going to see that evolution varies depending on what protein you're looking at. So, when you are doing this protein evolution, the thing you will first look at most of the times is what we call primary structure. And if you remember, primary structure is just the sequence of amino acids. So what we're looking at here is the primary structure of the protein cytochrome C from a bunch of different organisms, all the way from mammals to insects, fungi, plants. Now, when you do this overlap, you'll see certain things. First off are invariant residues. So if you look on the x-axis, it'll tell you at each position how many different amino acids were found there. If it's an invariant residue like this Q, this H, T, or C, you'll see a number 1. What that tells biochemists is that if that residue has never changed throughout the course of evolution, then that residue must be important, uniquely so, for the function of the protein. For example, here we have 4 invariant amino acids in a row, QCHT. This means that QCHT is critical for cytochrome C to work. No other protein can fit that position, or amino acid can fit that position. And if QCHT does get mutated, that organism probably dies. Now, from there, another class of amino acids called a conservative substitution. That is, we see substitutions by looking at different organisms, but they are the same class of amino acid, or have the same properties. For example, here we see there's 2 amino acids, but it's either E or D. It's all your negatively charged amino acids. Right next to it, we have 4 amino acids, but they're all nonpolar. So this means that it's not so much what amino acid is there, but rather that you have the characteristics there. So for the D and E example, it's really important to have negative charge there. For the example right next to it, it's important to have a nonpolar there. And then we have a hypervariable region. So these regions where you have a lot of mutations, and for that reason, biochemists think it doesn't really matter all that much what is there. So if we look at this amino acid, these 2 positions, we have 5 different amino acids taking that place. And if you just scan up, you see a lot of different colors there. Here we have lysine to negatively charged, so positive to negative, which says for this cytochrome C to work, it really doesn't matter what amino acid is there. And there's also something called neutral drift, which is just random mutations, but it doesn't alter the function. So maybe that 5 we were looking at, that K and E, you might actually call random or neutral drift. You would have to do more experiments to look at that, though. You can't really just tell from primary structure, is it hypervariable or just neutral drift. You will need to know how the protein functions and what positions are important for catalytic activity. By doing that type of graph we just saw, we can actually make a phylogenetic tree now. And this just tells us how organisms are related based on their proteins. So this is actually the phylogenetic tree for cytochrome C, which is the graph we were just looking at. And this was made by a computer. This isn't something a human sat down and made. They gave the information to a computer algorithm. The algorithm spit this out. And what you're seeing here is evolutionary tree. So each branch point is just saying that you have a common ancestor. And these numbers say, out of 100 residues, how many differences would you expect from your ancestors? So if you look at the mung bean to this common ancestor, there's 5, 2 for sesame. Kangaroo to this common is 2.5, so on and so forth. But you can see there was just one common ancestor, and then we branched off to our different types of organisms. And it's interesting when you look at this, the classifications of organisms make a lot of sense. Remember, this is just based on a protein alone, not looking at any other information, but you have mammals next to birds, amphibians, and reptiles, but not close to plants, not close to fungi, which is another piece of information that backs up the theory of evolution. Now, talking about protein evolution, scientists have sequenced over 50 million proteins now and have put those online. And using computer algorithms, we can now tell how related proteins are. Even if their sequence is less than 20% the same, these algorithms can still look at similarities between proteins. And what we have seen after we've looked at all these proteins is that certain proteins have what we call domains. That is 400 to 200 residues that don't change that much. Don't change structurally, I should say. A domain can have different amino acids, but it has a specific 3D shape to it that really matters for us. And so far we have cataloged over 1,000 to 1,400 protein domain families, although half of the proteins, or half all the domains we know, fall within 200 of these families. There's a lot, but a lot of those families only have a few members. Only 10 to 20% of these domains are just occupied by one protein. Now, usually, if a domain has greater than 40% identical sequence, so if you're comparing A and B and they have the same domains and have more than 40% sequence, they usually do the same thing. If the sequence is less than 25%, the domains might do different roles. Now, by looking at domains, looking at evolutions, we can start to group proteins into families, and we have special words for that. So if two homologous proteins have the same function but they're in different species, we call those orthologs. If we see it within a species, though, we usually assume that's due to gene duplication. So let's just go over what gene duplication means. So one chromosome will acquire both copies of a gene. So usually you have two chromosomes for a diploid. Each chromosome has one copy of the gene. And it's possible that a chromosome will get double the copies. Now, during this duplication, since you're producing an extra copy of a protein, this protein might diverge due to mutations. So there's no pressure for this extra copy to remain pristine, unmutated, if the organism already has a working copy and it has enough protein being made. So this other protein that has been duplicated is free to mutate. And we call this paralogs if you have homologous proteins within the same organism. Now it is possible that when you do this duplication, the gene just gets mutated and doesn't work. We call that a pseudogene. That is, non-expressed gene relics, and humans have a lot. But if you look at prokaryote, 60% of all proteins in prokaryotes are paralogs. We see copies of that in other proteins. 90% in eukaryotes and 98% in humans. So the majority of our proteins are in our own body aren't unique. We see domains in one protein in another protein in our body, which is suggesting that a lot of our proteins are due to this gene duplication event. We duplicated a protein, it mutated, and it got another function that made our ancestors better able to survive or more fit. Now, when you look at rate of mutations, it really varies based on what protein you look at. So histones. Histones are the proteins that wrap DNA, and you can see that it does not mutate much. So this graph is number of amino acids changed per 100 sites versus evolution time. And so histone is so well perfected at doing its job, this graph suggests any change to a histone will inhibit its ability to pack DNA, and that organism doesn't survive. If we look at hemoglobin, though, hemoglobin binds and transports oxygen. It can actually have mutations. It has some tolerance for mutations. And scientists have actually made a tree to show how the globin family has evolved over the years. Globins carry oxygen. We have myoglobin storage, but we also have hemoglobin, which is made out of this alpha and beta subunit, and that's for transportation. And the alpha, beta, and myoglobin look very similar to each other, but they have very different roles. And so the idea is that they have come from a ancestor protein. Now, if you look at another protein called fibrinopeptides, these help with blood clotting, and there seems to be no selective pressure for these proteins. You have a lot of mutations, which says that these peptides can still do their job regardless of the amino acid sequence in there. Or more, I should say, only a few amino acids really matter for fibropeptides to do their work, and the rest don't really matter all that much. Now, when we look at proteins and we're thinking about these domains, we find that proteins usually aren't just one domain, but they're multiple domains. So many proteins have multiple domains. And they can be different domains, like fibronectin. We have different domains here, which are these different shapes. Or they can just be a domain repeated over and over and over again, which we also see in fibronectin, where we have this one domain just being repeated a bunch. And what's interesting about domains is that they generally will evolve independently. You can think of domains as little mini proteins connected to are just held together by peptide bonds. And so as long as a little mini protein or the domain is still doing its job well, then the protein will still function. So they're almost like independent entities that can mutate on their own. So maybe one domain can tolerate mutations than another domain. So the domain that can tolerate mutations will mutate more frequently than your other domain. Now, when we're looking at domains and sequence identity between homologous domains, you're probably not going to get a one-to-one match. And that's because a lot of the time shape matters more than actual sequence when you're looking at different proteins. And although we do know a lot about individual domains, we don't always know what they do. That's because a lot of domains, or some domains, may not actually be doing biochemistry directly, but they're important for just spacing out the molecule just as a space filler domain, or they could be important for positioning everything in the protein so that the protein or enzyme is able to work correctly, and that's scaffolding. And that's it for this video, and I will see you in the next video.